everyone, this is Mrs. Mancuso, and this is an AP Bio video on photosynthesis. Today's focus is going to be going through the light reaction. So the last video that I made was an overview of photosynthesis, and that taught you that there are two major reactions, the light dependent reactions and the light independent reactions. This one is going to be focused on the light dependent reaction. So we call this the light reaction. So in order to have the light reaction, you got to start off with something that can actually absorb light. So we call these photosystems. So photosystems are clusters of pigments. The pigments could be chlorophyll, that's the most abundant one, or they could be accessory pigments. So there are xanthophylls and carotenoids. There's other pigments that are capable of absorbing different wavelengths of light, and they're not green. The the chlorophyll ones are green, but not every pigment is green. Now, because chlorophyll is by far the most abundant, it's green. So when you are green, you are reflecting green light, and that means you're absorbing the opposite colors. So other colors that chlorophyll is able to absorb would be blues and reds. So I'm going to show you a little spectrum so you see what I'm talking about here. Here are the two main chlorophyll molecules, chlorophyll A and chlorophyll B, and they're not really absorbing in this green range, but they're absorbing much more in the red ranges and purple ranges. And sometimes you will see an absorbance spectra versus a photosynthesis spectra. And just remember that if it's able to absorb light, it's probably going to be able to do photosynthesis. It's going to use different pigments to absorb the light. And it's not only based on chlorophyll, there are accessory pigments as well. Okay, and then this is important to understand too. The wavelength and the intensity of light that a plant is exposed to is definitely going to affect its rate of photosynthesis. So I want you to picture blue light versus green light. If for some reason the wavelengths of light that are available are more in the blue range, as long as your plant is green, it's probably going to do well. But if instead that plant has bluish hues to it and it reflects blue light, it's not going to be able to do as much photosynthesis or make as much sugar because it's reflecting the light instead of absorbing it. And then the intensity, that's pretty easy to think about living here in Rochester, because you can easily think of days when sunlight is very intense. So that means that a lot of light is available to go into the plant and be absorbed, and that energy used to eventually make sugar. And then there's other days when it's really overcast and cloudy, and it just seems like a plant would never survive on those days because it definitely wouldn't be absorbing energies of light. All right, so let's move on to photosystems. Photosystems, I like to think of them as a big clump. And there's all these accessory pigments around the outside, and then the middle is called the reaction center. And the reaction center is made up of chlorophyll A molecules. The accessory pigments are able to take light that's coming in and channel it to the middle. In the middle, those chlorophyll A molecules are going to be excited. And when they get excited, if you remember the absorption of light in chemistry, they actually have their electrons not just jump to a higher orbital, but they can actually go right out of the chlorophyll. So when they go out of the chlorophyll, it loses one electron each for a total of 2 electrons. So I have that written right under here. The absorption of energy, so we're talking about light energy here, it excites 2 electrons and sends them through an electron transport chain. And we'll draw an electron transport chain and make sure you understand it. As those 2 electrons lost from photosystem 2 go through an electron transport chain, they lose energy as the energy moves. So think of like a hot potato going through. It's going to lose energy as it's passed, and the same thing happens with electrons. But what happens is the energy gets used to move things from an area of low concentration to an area of high concentration. So it's able to pump hydrogen ions against the gradient into the thylakoid space. So I want to spend some time drawing this out and making sure you understand. So first I'm going to draw just one part of a grana. So this would be one thylakoid membrane, and inside of here is the lumen. And I'm going to start way over here. This is photosystem 2. So photosystem 2 is the first photosystem that light is absorbed by. So I'm going to have light coming in, and light is absorbed by the photosystem. And inside of this photosystem, there are my 2 chlorophyll A molecules, and they get excited. And when they get excited, they lose 2 electrons. And when they lose the 2 electrons, the electrons are actually transferred, and they go to another molecule. So this would be another protein. It makes up part of the electron transport chain. You could call it an electron acceptor. It's the first thing that an electron moves to when it leaves. So an electron acceptor is going to take those 2 electrons and keep passing it. Now, as they're passed, usually the next thing it encounters is a pump. So this right here would be a proton pump. I want to use a different color here. All right. This proton pump is able to move H plus into the lumen. So H plus is going to build up inside of here. Okay, and the reason why they came across, this is a proton pump, so it'll move them from high to low concentration using energy. The energy is coming from these 2 electrons. So the electrons keep moving, and eventually they end up with another photosystem. So I draw the photosystems really big. This is photosystem 1. Photosystem 1 also gets excited by light energy, and that sort of reactivates. Here's my electrons coming through. It re-energizes my 2 electrons. All right, and they continue to be passed to another electron acceptor, and eventually they go to, I'm just going to get this out of the way for a minute so it's not blocking my space. Okay, eventually these electrons are going to end up in a molecule that is known as NADP reductase. I'm going to type that. NADPH reductase. Okay, the ACE tells you it's an enzyme. So this molecule, what it's able to do is it's able to take, I don't want that color. Yeah, let's use red. Okay, NADP plus. All right, if you take NADP plus and you reduce it, reduction in chemistry is, remember oil rig from chemistry? Rig, reduction is gaining. So these 2 electrons are going to go with the NADP plus, and you're also going to add a hydrogen ion, and that's how you form NADPH. So NADPH is one of the products of the light reaction, and that is formed from these electrons, which originally came from back over here from my chlorophyll, but they got excited by light. They went through an ETC, an electron transport chain. They got re-excited at photosystem 1, went to another electron acceptor until eventually they ended up with NADPH reductase. Now that reductase reduced NADP plus. So NADP plus is here in the stroma, and it combines with 2 electrons and 1 H plus. And if you think about it, H plus and a plus on here become neutralized by 2 electrons. So the 1 negative balances that, 1 negative balances that. You end up with NADPH. NADPH is a large molecule. It's a coenzyme, and it's going to help in the reactions of the Kelvin cycle. So this is one step of the light reaction. So now I want to go to this next drawing, and you're going to be like, oh my gosh, Mrs. Mancuso, seriously, look at how neat their drawing is, and yours was a disaster. Okay, yes, mine was a disaster. So, but it's great at showing, once you've drawn it yourself, then it's great to look at diagrams that are capable of doing these lovely diagrams, and then you can label them. So here's my photon of light coming in. And here is my first photosystem. So this is photosystem 2. And it's able to lose 2 electrons. And they are going to go to an electron acceptor, which is this molecule here in the middle. And this would be a proton pump. And it's a pump, which means that it's moving things from low to high concentration, and it's using some of the energy from the electrons. And then this one doesn't show it. I need another diagram to show that that transfer keeps happening. All right. This one's only showing part of it too. So again, you'll have to grab Google, really, and get some more diagrams to show you more steps and take you through both photosystems. I'll make sure that you get it all in your notes. All right, now the second part of the light reaction, so the first part was all about producing NADPH. The second part of the light reaction is about splitting water. So splitting water is called photolysis. So photo means it uses light energy. Lysis means it breaks apart. And it's definitely a type of hydrolysis because it's using water. So water is going to be split inside the thylakoid space, or what I call the lumen. When the water is split, this is what you get out of one water molecule. You get two H pluses. Those are going to build up in that thylakoid space. So if this is my photosystem, here's water right here, H2O. And when the light comes in, it's able to break that water into these things. So it's able to break it into H pluses, and the H pluses build up here in the lumen. It's going to make oxygen, and the oxygen it makes is going to go right out because it's going to move from high to low. And eventually it's even going to get out of the leaf, not just out of the chloroplast or out of the cell. It can actually get out of the spongy mesophyll area and escape through a stomate. Or it could get used by a mitochondria to do respiration. All right, and then the two electrons that are broken as water is broken, they are going to go replace the two electrons that photosystem 2 lost. So that is a really common question and a really common idea. What happens to those electrons that photosystem 2 lost? Water replaces it. Water replaces the electrons that were lost. All right, let's see here what comes next. Okay, here is a diagram. Now this one is showing you things a little bit reversed. So here's my lumen, here's my thylakoid space, and then out here, this is the grana. This is the whole thing makes up a grana. This is the thylakoid membrane. All right, and here's a photosystem, and here's the splitting of water. So the two electrons go to replace the electrons that chlorophyll A lost in the reaction center. And then the H pluses build up in this lumen, and then the oxygen's going to head on out. All right, and then the electrons again, plastoquinone is the first electron acceptor. You don't have to know all of their names, but that's why you're going to see PQ, or this one's plastocyanin, but these make up our electron transport chain. And this, of course, is the proton pump because it is going to move H pluses from low to high. And this arrow should only be going this way because that's what a pump does. All right, so when you have built up these H pluses inside of the thylakoid space, you actually create a gradient that you're going to be able to use. It's sort of like building up water using a dam and then letting the dam go, and now the water had so much potential it provides energy. So the buildup of H plus in the thylakoid space causes a gradient. You could call it a proton gradient or an H plus gradient. You could call it an electrical gradient because it's definitely a difference in charge. It's also a difference in pH because H plus is how you measure pH. So you've definitely built up a gradient, and these words can be used interchangeably to describe that gradient. All right, gradients can be used to create energy. Think of like a windmill, a watermill, a dam, and there is a new word in red here, chemiosmosis. Chemiosmosis is utilizing a proton gradient to make ATP. Using a proton gradient to make ATP. Now, there is a special membrane protein. It has a funky look to it. I'll draw it for you. It's called ATP synthase. it's going to use the H plus gradient to add a phosphate group onto ADP. So I'm going to show you how that happens. Oops, sorry, I want to just create an open page here. Okay, and again, I'm going to do my diagram first, knowing that you're going to be looking at other diagrams that are going to be much neater. But here is what it looks like. It sort of looks like a channel, but it has a spinning rotor on one side. It's always the side out of the grana. So out here, this is the stroma. So my rotor is going to be facing the stroma side. In here, this is the lumen, and this is where all the H plus is built up, because as the electrons went through, the pump pumped more H pluses in, and water was split, and that added to the H pluses. So here's my proton gradient. These start moving through, and what happens is attached to this, ADP and PI. And as this spins, it actually attaches it to it. I'm just going to have it go down like this, and it makes ATP. So ATP synthase is what we call this channel pump. Sorry, it's a channel for the hydrogen ions, but it is a it has a spinning end to it that makes it like an enzyme. So ATP synthase. All right, and I want you to add. Well, you already added this to your notes. At the end of the electron transport chain, NADP plus gets reduced to form NADPH. So we took the NADP plus, and we took an H plus, and we took the 2 electrons, and we made NADPH. Okay, here's the diagram I was looking for before. So I know it might look a little bit overwhelming right now, but I want to spend some time digesting it with you. So here is my thylakoid membrane, all right? Inside of here is the lumen, and outside here is the stroma. This is photosystem 2, which is also called P680 because it absorbs 680 nanometers of light very well. That's the wavelength of light that it absorbs. And here's light coming in. And then down here, this is photolysis. This is the splitting of water that happens right at photosystem 2. When you split it, you get oxygen. That's going to go out. And you have H pluses. This is what builds up our gradient. All right. Then you have 2 electrons, and the electrons go right here to replace the electrons that got excited and went to the electron acceptor. So as light comes in, 2 things are happening. Water is split, and electrons are lost from chlorophyll. The electrons from the water splitting replace the electrons that were lost so that photosystem 2 isn't missing electrons. All right, the electrons go to an electron acceptor, and then they go to a pump, and they keep on moving until they end up at photosystem 1. Now, this pump allowed H pluses to come in, and as H pluses come in, they're going from low to high, and they're using the energy of the electron flow. All right, photosystem 1 gets their electrons re-excited, and then eventually these need to be drawn a little bit more in the membrane instead of out of it. But this is my reductase, and this is what's able to take NADP plus and add it to an H plus and add those 2 electrons and make our first product. So there's NADPH, one of our products. The second product is right up here. So ADP and phosphate use the channeling of H pluses through to spin this rotor and make ATP. So that's our other product of the light reaction. Okay, products. NADPH, that was made from the electron flow. That's needed for the Kelvin cycle, which is the next reaction. ATP, that's also needed for the next reaction, and it was made due to the buildup of the H pluses. The last product was oxygen, and that's what can leave the chloroplast, can leave the cell, can go out of the plant through the stomate. All right, so that's the end of this video. Again, take time to go back through it, draw it all out for yourself, Google some images, and study.